Hello, my name is Grant Johnson. Today we're going to be learning about action potentials. So, in your nervous system you have a cell, and on that cell it has what is called an axon, which is basically just an extension. And then over here you'd have another cell receiving that signal. So how do the cells actually generate the signal? Well, it has to start here at the axon hillock. And this is where you can have two things occur, either a temporal stimulation where you have a bunch of signals at once, or a summation of signals from other dendrites that are receiving signals.
What are dendrites?
So this is the axon, which sends the signal. On every other cell there would be dendrites that are going to receive the signal at the synapse, but that's a completely different.
that looks like a unicorn with a fat ass.
So, to get this signal started, your cells actually make electricity. And they do that by having a difference of essentially protons on certain sides. And so there's two ions that are moving, potassium and sodium. Now there's these pumps inside the membrane of the cell that are going to allow this to occur, and they do it by having a counter flux. And so they actually send 3 sodium out at the same time they put 2 potassium in. So you can see if they're both positive, you're going to have a net positive effect out here, so the cell will be more negative. Now since it's more negative, you're going to have essentially a signal here at this point, and it's going to look like a wave. And so, basically, the threshold of the membrane is normally around there, negative 80 millivolts. And so, in order to send a signal and start the response, it actually needs to get to about negative 50. Or sorry, 65. And so it's kind of a range, but basically, eventually this has to become slightly less negative. If this is negative 80, this is negative 55. Going up on the graph means you're getting more positive, and that's because you have 3 sodium moving out, and then suddenly you open the gates and you allow the sodium to come back in. So as soon as it hits negative 55, you open the sodium gates. Sodium floods in, it's positively charged, you get higher, and if you hit the peak, then you're going to have all the way to the rest. So if this occurs, this negative 55 threshold right there, you're going to send a signal. And it's basically just going to travel along this axon the whole way. The axon is covered in myelin, which is basically just fat, and it acts as a conductor to increase the speed that it goes. Do something called saltatory conduction, which in Latin just means to jump. And so basically, the electrical signal jumps in between the myelin, the fat, and it goes faster. So certain things like Parkinson's, multiple sclerosis, certain nervous system degenerative diseases actually remove this myelin, and then you slow down the process. And so basically the whole thing is moving through electricity, through the influx of sodium, and then eventually the potassium gates will close and things will equal out, and you'll get back to your resting membrane potential when you're not sending a signal. But the whole thing is controlled by ion gates. Once you get all the way down to the synapse, things get a little more interesting. So if you have the end of the axon reaching the next cell, what we call the postsynaptic cell, then it's going to continue to pass on the signal. So basically the electrical signal from that sodium influx is going to go all the way down here. It's going to stimulate certain proteins to form and vesicles to bud with the membrane and release their contents. And what their contents is usually a neurotransmitter. Things like serotonin, dopamine, acetylcholine, things that like drug pathways interfere with. This could just as well be nicotine. And so a bunch of things are flooding into the cell, and then it's going to have a receptor to catch them and send the signal on. Now, for this vesicle to actually bud, you have to have an influx of a different ion, calcium, that's also positively charged, but again, just carries that electrical signal from the sodium. Calcium will bind with these vesicles, signal exocytosis, and then the vesicle buds off, and you send the signal on. So the whole thing is basically like electrical, and then here it becomes chemical. And the reason that's important, because it's really easy to regulate this. You have enzymes that come in and will eat up whatever neurotransmitter this is to stop the response. And so you can regulate that. And a lot of drugs that we take come in here and maybe bind to receptors to prevent the chemical. That'd be like an antagonist. Or maybe other drugs are inhibiting this enzyme, and then you have this stay in the synapse for longer, and that would be an agonist because it increased the response. You can have a lot of drugs that do these opposite effects by like binding to different things. Ultimately, that regulates it, because if we didn't have that, we'd have like a type of electrical signal that's just in a muscle, and sometimes you can like experience a cramp. And that's kind of like a dysregulation of that signal, because it's really hard to cut off electrical signals once you send them, unless you convert it to some chemical response along the way.
What the fuck?
Any questions?
Dude.
I have a question.
Yes.
One question. In the first part, when you were talking about the sodium influx,
Yes.
and the resting state is negative 80 millivolts.
Yeah, roughly. Yeah,
Roughly.
I think so.
So within the cell, there's always it's always negative. There's more what is it called? Negative ions.
Yes.
And positive ions.
Yeah. You can think of it that way, or you can think of it as there's less positive ions.
Less positive ions. So what does like
I think it's actually like negative 70 millivolts. It doesn't actually, and then the membrane potential to actually like start the response is negative 55.
So the reason why it would break the threshold can be a million different things.
It's usually from receiving a signal from another cell.
But that signal must have started somewhere.
Yes, and so that's where you would be thinking of something like the origin of consciousness, and like where do like original neurons fire without receiving signal, which is also kind of tricky to think about, because your body's always receiving signals from your environment, right? Temperature, pressure, pain, things like that. Even like your auditory neurons are firing right now, hearing the vibrations in the air, and that's what's receiving signal, allowing you to hear me. And so there's always a sending of signals, and like I said, when you talk about the axon hillock, the reason this part where like the body, what's known as the soma, because this is a cell, right? It's it's a cell, but it's a specialized cell that sends a signal, and they have to basically, the signals it receives from other cells in its dendrites have to either like supersede a point here in a temporal fashion where they're happening, let's say this happens, and this happens simultaneously. So you receive enough signal to then pass it on. But if this happens, a second later this happens, a second later this happens, it might not generate enough internally to reach this point and send on the signal.
Break the threshold.
The axon hillock, yeah. And reach the threshold, exactly.
Huh.
Does that make sense? Because maybe they diffuse out of the cell.
So if you are lacking in potassium, for example, does it hinder with this process?
It could, yeah, it definitely could. You don't really need a lot of potassium anyways. Like, it's very rare to be just in that micronutrient. You see what I'm saying?
Right.
But you're right, because it's the sodium, potassium pump.
I have a stupid question.
Yes.
What about iron?
So iron is important because it binds to hemoglobin within your red blood cells, and it allows it to carry oxygen.
Is that our next lecture?
That's
Well, just say
that's
say-
actually really crazy because, dude, hemoglobin is like insane for a lot of reasons.
Okay,
'Cause I
say
think
next
I have,
time.
I think
Next
I have an iron problem.
Really? You
Yeah.
should. Do you take supplements?
No.
Okay, I would take supplements, and I would try to eat like red meat, like once or twice a week too, so you can get natural.
Yeah.
Like the
I'll
sliders
show
at
you my
work.
calves, yeah.
Your
The
calves.
sliders make me.
You don't like the sliders.
say,
Then
say
Anywho,
on our next time.
on our next time, we'll be talking about hemoglobin in red blood cells.
this is Grant.
This is Grant. Thanks for tuning in.